Greetings everyone. Today I'll be presenting my research project on artificial in vitro blood-brain barrier model for preclinical screening. Now, for drug development for CNS diseases, the major problem is the blood-brain barrier. The blood-brain barrier is an anatomical feature of the CNS, which provides protection against any drug or any large molecule to enter the brain. Now, for this reason, the major problem in development of the CNS drugs has been seen. Now, which creates a difficulty for the drugs to pass through the BBB. And for this reason, the in vitro blood-brain barrier models have become very useful as an alternative to the in vivo models. Now, there are many multiple models present, such as in silico, in vivo, in vitro, and artificial membrane-based models. Now, to give the background of my work, first I'll talk about the xyloglucan and P-glycoprotein. So, xyloglucan is a member of a polysaccharides group referred to as hemicellulose. Now, xyloglucan-based hydrogels basically exhibit very high water uptake capacity, swelling ability, and gel capacity, which makes it very useful in the tissue engineering. Now, it can be used because of its control of cell expansion property and its biocompatibility. Next is P-glycoprotein. So P-glycoprotein is a multi-drug resistant protein and also a member of the ABC transporter family. It is abundantly present in the BBB membrane and other barrier cells as well. Now, P-glycoprotein being a efflux transporter creates a very limiting effect, and it is also a very major problem for any drug to pass because it gets effluxed by the P-glycoprotein. So the rationale is that permeability of any drug or drug delivery system is the most important factor through the BBP for the drug development process. Now, for such models, xyloglucan-based hydrogel membranes can be used for upregulation of tight junctions and decrease in the parallel cellular permeability and also to improve the barrier properties. Next, the cell lines expressing the higher efflux transporter proteins can also help in mimicking the pharmacokinetic properties. All in all, that can make a very high throughput, cost effective, reproducible model which can be used for the predictive permeability of the drugs. Now, the objectives of my research are first the optimization, preparation, and characterization of a xyloglucan-based hydrogel membrane. Next is cloning of the P-glycoprotein gene and preparation of the stable cell line, followed by the development and characterization of an artificial in vitro BBB model. So, first is the optimization of the membrane. So, multiple factors were considered, such as crosslinkers to be used, concentration, temperature, and the time. And after using multiple crosslinkers, physical and chemical, checking different concentrations, different time ranges, and temperature ranges, finally citric acid was chosen. And as you can see here, citric acid along with xyloglucan can form this crosslink structure. So this is the optimized parameters. So polyvinyl alcohol was used as the plasticizer, xyloglucan as the gelling agent, and citric acid as the cross-linking agent in following concentrations. So xyloglucan is dissolved in the water along with citric acid and then polyvinyl alcohol. Now this solution is poured on the Petri plate and the films are let to dry for overnight at around 45 degrees Celsius and then shifted to vacuum at 90 degrees Celsius for around 16 to 18 hours, after which the membrane is cross-linked. This is the image of the membrane that is being made. Next, the characterization of the membrane was done first using FTIR analysis. In the FTIR analysis, we could see the ester peaks confirming that the cross-linking was actually done. Next, the characterization was done based on the mechanical properties. So for the film, the toughness and the strength is very important since it will be used in the cell culture. And the film containing the plasticizer showed very high strength and toughness. Next, the swelling index was tested. So the film swelled around 105% in around 24 hours, but the film did not disintegrate in the water, which is what the crucial point was. Then thickness analysis was done and the mean thickness was found. Because of this low standard deviation, we could see that the film was very uniform in thickness throughout. Next, Scanning electron microscopy analysis was done to check the pore size of the membrane. Since if the pore size are too long, the cells can easily leak through the membrane. But the membrane had pore size in the ranges from around 4 to 7 micrometer, which was the ideal range, and the cells can adhere on it very easily. Then sterility and the biocompatibility of the membrane was tested. So the membrane was sterilized using UV radiation and no contamination was seen. Also, MTT was conducted and cell proliferation was around 89.53% after 48 hours, confirming that the cells are not, were easy to grow and the membrane is not toxic to the cells. After this, the cloning of PGP was done. So from this construct that was ordered, it was isolated and the plasmid was isolated. Now from this plasmid, the PCR was done to amplify the PGP gene, which was then isolated. From this isolated gene, the restriction digestion was done. And these are the digested products. So the digested gene and the digested plasmid were then ligated using DNase ligase enzyme, and then it was transformed in the bacteria. So these are the colonies that was observed, which were then screened using colony PCR, and then the screen colonies was then isolated and sequencing was done to confirm the clone. Now, to check if the clone is actually effectively working inside the cells or not, it was transfected inside the cells, and by QRT-PCR, the RNA levels of the PGP were analyzed. So we can see that the RNA levels were quite high for PGP in the cells that were transfected with the clones, telling us that the RNA was being made inside the cells for PGP. Then similarly, using Western blotting, the protein levels were checked for P-glycoprotein, and we could see the bands, stronger bands as compared to control, showing that the cells were expressing P-glycoprotein in higher amount than the control. Then functional assay was done to check if this protein that is being expressed is actually functional inside the cells or not. So rhodamine fluorescent dye was used, which is a substrate for PGP, and that gets effluxed out of the cells. So as expected, the fluorescence levels in the overexpressed PGP cells was very less because the rhodamine was effluxed out in these cells, and this was also confirmed using fluorescence microscopy. Next, PGP stable cell line was formed by transfection using lipofectamine, and then it was then selected for around 4 weeks using blastocidin and antibiotic treatment, and a change was in morphology was observed. Next, the characterization was done using same RNA expression levels and protein expression levels, both of which confirmed that the stable cell lines was expressing the protein and the RNA for PGP at a very high level. Now the in vitro model preparation was done. The transfer was coated with the membrane and then cells were seeded on it. Then the evaluation of the barrier function was done using tear measurement, and around 750 ohm per centimeter square was the value for the model prepared using the stable cell line, which was very high compared to the CARCO2 used model prepared using CARCO2 cell lines. Next, the permeability assay for the drugs was done, and the permeability coefficient was calculated using this following formula. So for donepezil, which is a high permeability drug, as was expected, the donepezil permeated through the model very easily, and the flux rate was also higher in the stable cell line containing model, which was expected. Then in carbidopa, which is a low permeability drug, the permeability was low in the cells, and in stable cell line based models, it was the lowest as compared to the control, which was CARCO2 cells. Then for the atenolol is the drug which is usually effluxed out by the PGP. So the atenolol was also checked, and the efflux and the flux rate for the atenolol in case of stable cell line was the lowest as compared to normal CARCO2 cells and vinblastine-induced CARCO2 cells. All in all, this could validate the model that the stable cell line is functioning well, and the model is also showing the flux rate as expected. So to conclude, I can say that the prepared model is very easy to use, economical and highly scalable, since it includes only one cell line to be used, that is the stable cell line, which is also mimicking the pharmacokinetic properties, and xyloglucan based membrane also, which provides the enhancement in the tight junctions. So it is a very simple to use model, which is highly scalable, highly economical. And as a future perspective, the prepared stable cell line and the model can be used for drug testing for BBB as well as for intestinal and other barriers as well. Thank you.